All right. Welcome everyone to the MGB Neurology Grand Rounds today. My name is John McLaren. I'm one of the current child neurology chief residents here at Massachusetts General Hospital. I've included the link and code to claim CME credit for today's conference in the group chat. If you have any questions throughout the lecture today, please direct them to me in the chat or in the Q&A portion of your sidebar, and I'll read them aloud at the end, time permitting. My honor today to introduce our speaker, Dr. Albert Misko, instructor of neurology here at Harvard Medical School and one of the things. After graduating from the University of Florida in 2003, Dr. Misko completed his MD PhD at Washington University in St. Louis, followed by 5 years in the pediatric neurology residency here. He completed his R25 studying sulfite-mediated inhibition of aerobic respiration and sulfite oxidase deficiency, where he won the Child Neurology Society's Young Investigator Award. And this was followed by a K-12 researching sulfite-mediated neurotoxicity and sulfite oxidase deficiency. As an instructor in neurology here, Dr. Misko specializes in the care of children with inherited neurometabolic disorders. As a clinician scientist, he's building a translational research program that develops genetic medicines for the patients he has the privilege to treat. Dr. Misko's research program strategically focuses on rare monogenetic disorders. The ability to rapidly identify a single genetic variant driving a disease process expedites the development and application of genetic medicines in human patients. The high-level goal of this research program is to leverage this unique situation and create a rapid, synergistic exchange of information between translational and reverse translational efforts. But to this end, Dr. Misko's group harnesses emerging genetic technologies to create novel gene therapy strategies, collaborates with industry partners to de-risk the application of new gene therapies, and conducts gene therapy trials to gain insights that inform the next iteration of his group's research efforts. Without further ado, Dr. Misko, the floor is yours.
Great, thank you so much for that introduction, John. And a big thank you to all of you for considering me to fill in for Kathy. It's a huge thrill to be here giving grand rounds, and I'm very grateful. So my talk is going to focus on developing gene therapies for these ultra-rare diseases, and especially in the academic setting, purposefully. These ultra-rare diseases are such that the low prevalence is not profitable for many members or all members of industry. And it really falls on, I think, academia more to fill in and try to bring meaningful treatments to these devastating disorders. Rather than concentrating on the technologies themselves, like the mode of gene therapy delivery or the mechanism of gene editing, I want to focus on some of the other challenges that come around forming these technologies and these strategies here in academia. How do you define the natural history of the disease? How do you find the patients? How do you understand the disease pathophysiology and build up to the biomarkers in order to facilitate the trials and the first in human applications? So these are my disclosures briefly. In order to start this discussion, I'm going to really build this around two projects that our group has been working on heavily. The first is an ultra-rare disorder called sulfite oxidase deficiency and molybdenum cofactor deficiency. Sulfite oxidase is a molybdenum cofactor dependent enzyme, and these diseases are clinically, radiographically, and pathologically identical. So I'm going to group them together for the sake of discussion. It makes it easier to facilitate about these disorders. And collectively, they're referred to as the sulfite intoxication disorders. There's a bit of buzz right now in the pediatric neurology community around molybdenum cofactor deficiency type A, as there was recently the first FDA approved drug to treat that subtype in order to extend disease or in order to extend survival in those patients. And that was something developed originally by Günter Schwarz in the University of Cologne. And it was a long time in coming. He was one of my mentors, and it was really cool to see. The second story I'm going to tell you is about mucolipidosis type 4, which has a very long legacy here at MGH, which we'll talk about. And when we talk about sulfite oxidase, that deficiency, that project, I'm going to sort of talk about it from the bottom up about pathophysiology to gene therapy to patients, whereas with mucolipidosis type 4, we're going to focus more on the natural history and how to bridge that gap from a working preclinical treatment to that first in human application or that proof of concept application. In order to address these disorders and to start the whole discussion, I just want to kind of take a step back and why ultra rare disorders? Why only target these small groups of patients? Well, as physicians, when we approach these really complex diseases, actually any disease, right? What we're really thinking about is the person or the system as a whole. We all as organisms are self-organizing, decentralized, complex systems. And now after years and years of research, we finally understand the nuts and the bolts, and that's the genes and the proteins, right? And now it's sort of the onus is upon us to take those nuts and bolts and figure out how they come together. And that's the new systems biology sort of way of thinking. How do these things come together? What are these nonlinear dynamics that define these emergent properties that come up at all of these different levels of organization? Now, it's true when looking at all sorts of diseases, right? You can go from top to bottom and think about how all of these nuts and bolts, like the genes and then the enzymes, subcellular networks, cellular networks, affect the higher levels of organization. But it's also true that these higher levels of organization have top-down regulation and also affect these lower levels. For instance, maybe if the mutation that you have is in a channel, that results in epilepsy, which changes the functional network architecture of your neural networks. At the same time, that enhanced abnormal synchronous firing feeds back down and changes in the metabolisms of the cells at that lower level of organization. So these are all fed in together. And of course, as physicians, we're always wondering, well, what's the therapeutic target? And it can land on any one of these levels of organization. For instance, on the functional network level, infantile spasms, right? This is a convergent phenotype that could be caused by structural injury, metabolic disorders, genetic changes, but the therapeutic sweet spot is ACTH. You see the spasms clinically, you see the hypsarrhythmia on EEG, and you know what to do to affect that disease process. At the same time, you have to try to figure out what's underlying it. If it's a structural abnormality, perhaps surgical intervention is enough to actually affect that disease process, all with the ultimate goal of what's here at the top, patient quality of life. And that's the ultimate goal. No matter what we tinker with on these lower levels, it's all to that ultimate goal. So the ultra rare diseases, and why to focus on these, is because they offer this really cool insight, right? Like, we know what's kind of going on here. The defects have been very well defined because some of these disease variants in these genes they're all risk factors. But in these monogenic disorders, those risk factors, it accounts for like 99% of the risk. So you know what to go after. And in fact, with these new treatments that are now available, all these AAV-mediated transgene delivery systems, since we already know the gene that we want to deliver, I mean, we almost have this ethical and moral obligation to try to do something, maybe even before we totally understand the disease pathophysiology and everything that's going on at those higher levels. By the way, this sort of thinking about systems biology is where Dr. Cavanis was before he left and retired. And I just want to point out that he was really thinking about this, and he was sort of pioneering this in his teaching. And I'll always hold that with me because he was such a huge part of my training. Knowing that these are the areas that are affected in these neurometabolic diseases and these monogenic diseases, we can have fun. It's sort of a playground, thinking about all of the different ways we can affect the disease processes at this lower levels with the hope of affecting the higher levels of organization. So for instance, with neurometabolic disorders, perhaps if there is an enzyme deficiency, we can use substrate inhibition to prevent the buildup of toxic metabolites that are not being properly processed. Perhaps we could do enzyme replacement therapy and give back an enzyme that is missing, or go on to the gene level, edit the gene, deliver a gene. Notice I have replacement in italics. I want to make this is a really important point. Gene replacement is never gene replacement, because these self-organizing systems, these human beings, they develop in the absence of that gene completely. You're not replacing anything because it was never there. Not like a robot that's missing a nut or a bolt or a piece that you're just putting back, but rather what you're doing is delivering an exogenous synthetic gene with the goal of causing a state shift in the target cells to a therapeutic end. And that's really important to keep that in mind. That's what we're doing, because developmental context is important. You have to consider what changes happen during development before treatment that may or may not be reversible. What epigenetic modifications, what adaptations have taken place that you don't even realize, and how much can you actually affect them? In fact, what we're really doing at this level is we're doing the first in vivo, in human genetic manipulation, genetic engineering in a very, very real way. And that has to be sort of appreciated. There's one other thing I want to bring up, or two other things about sort of thinking about disease treatments in these disorders, and that you also have to think about their evolution both in space and time, because that dictates the strategy and how you're going to develop it. Evolution in space, it's a lot about the intrinsic dependencies of a lot of different tissues on the gene that's being affected in these disorders. It's also important to note that there is cell autonomous and cell non-autonomous disease mechanisms in these disorders, meaning that sometimes a tissue is injured because it needed that gene or that enzyme, whatever the protein that's not functional. Other times it's because something that goes on in another tissue, perhaps the production of a toxic metabolite, can then go and affect another tissue that wasn't dependent on the gene originally, but is affected by that toxic metabolite. The other thing to keep in mind is evolution in time. And I was kind of think about the example of, like, you can hear things coming. You can hear disease coming. If you're at home, it's late at night, it's dark, but you hear footsteps coming down the hall. You probably already know who that is based on the prosody of their step, their cadence, how hard they're pressing on the floor, and that's all dictated by weight, height, and all these different characteristics of the person. In the same way, you can sort of hear these diseases coming. They have a different prosody in which they develop. And I was trying to illustrate that down here with all of these different sort of sound bites. Sometimes there's these precipitous sort of oscillating presentations. Sometimes they build slowly over time. How the disease develops in time is really important to think about. When do you want to treat them, and how is that going to affect treatment outcome? And I want you to keep that in mind too, because that's extremely important, the prosody of the disorder. Okay. So let's turn to the two examples then that I want to bring out here, both that sulfite oxidase deficiency and mucolipidosis type 4. And we'll start with the sulfite oxidase and molybdenum cofactor deficiency. I became really interested in these disorders as a resident when I had the privilege of caring for a few of these patients. And then through Florian Eichler, my mentor, I was able to see even more patients. And then working with a company called Origin Biosciences, I was able to see many more patients and reach out to the whole community internationally, which has been really exciting. Now, we knew a little bit about this disorder at the onset, right, just from case series and case studies. And what was fascinating about it was that this disorder seemed to mimic neonatal hypoxic ischemic injury. These patients were born, and within the first days of life, they had acute encephalopathy, refractory seizures, feeding difficulties, motor abnormalities, including movement abnormalities, rigidity, even some hypertonicity and hypercaplexia really early on. And eventually, with the sort of advent of MRI, we started to appreciate too that the ancillary imaging looked like hypoxic ischemic injury. It had been described before: T2 hyperintensities throughout the cortical ribbon, diffusion restriction, very large lactate peaks with preservation or relative preservation of the brainstem. So all of these things really sounded a lot like hypoxic ischemic injury. But we didn't have a lot of natural history. So the first thing that our group did is we turned to the literature, and we pulled out about 126 patients from the literature and extracted all of the clinical data available on them. And as a result, we were able to define both the onset of symptoms, the median is about 1 day, and the survival of about 31 months. We're also able to add to the literature and describe both typical, which we're going to talk about here, but there's also actually very cool atypical variants that present as movement disorders later in life. But turning to these are, I think, the really interesting ones here. So it took a literature search. That's the first thing. There just weren't enough patients with this ultra-rare disorder, but we're able to make meaningful insights by just taking a lot of time and really trying to pull out all of the data from the literature. The next thing we were able to do is, because Origin, which originally had been Alexion, invested so much time in this, they had collected about 23 cases and had a library of MRIs. And we didn't have enough longitudinal studies from any one patient, but we were able to take them, organize them by from onset of symptoms to when the study was taken. And then we kind of like compressed them into a single cohort. Now, this is not ideal, obviously, and you have to take all results with a grain of salt, but it was actually really cool. We were able to really delineate some of the longitudinal evolution of the disease with this technique. So with Paul Caruso and Karen Buch and I sat down and we spent a lot of time doing this. And I don't want to go over all of the details. I'm actually showing you more of this data just to show you how much work it was. But let me walk you through what we found, which was super interesting. We found that indeed, a lot of these images, the evolution of MRI abnormalities really did follow both the characteristics of neonatal hypoxic ischemic injury spatially and temporally. So forebrain was involved in most of these kids, and that's cortex, subcortical white matter, deep gray matter, with relative preservation of the brainstem and the cerebellum, which weren't affected acutely. And at the time of onset, again, these kids just had raging diffusion restriction throughout all of these affected areas, huge lactate peaks, a GLX shoulder on the MRS, which is basically elevated glutamate as a sign of excitotoxicity. It's seen in like the periictal or postictal setting. And it's also just all that glutamate that's spilling out into the extracellular space that's detected. And we were able to categorize these kids even into an acute, a subacute, and chronic phase, just because it was, when you're looking through the images, it just became very obvious that there were these different phases of evolution of the features. And indeed, those time courses, or these buckets of time, which were roughly about 0 to 7 days, 7 to 21, and then beyond 21 days, really sort of echoed the changes in the timing of evolution in hypoxic ischemic injury in the neonates as well. So while the forebrain was highly affected, the hindbrain wasn't, and it was relatively preserved, because these neurons, metabolically, ontologically, they're very different. They're a different beast. Their metabolism is different, and they were very resistant. Cerebellum, like in almost all disorders, right, degenerated at some point for sure. But one important part about this study was that we wanted to be able to provide clinicians with at least some features that might help distinguish these kids from hypoxic ischemic injury. And things that we found included, at the time of birth, some of these kids had a smaller atrophic corpus callosum, a smaller atrophic cerebellum, a very large posterior fossa, which really in most of these kids looked like a cisterna magna, or a mega cisterna magna. Not so much a Dandy Walker malformation as had been described previously, but really it's that mega cisterna magna. We also found that there was a very small fraction of kids that already had signs of chronic injury at the time of birth as well. And then putting all of those things together, it was not definitive pathognomonic signs, but definitely radiographic biomarkers that could help signal the need for further workup for these disorders in these kids, especially now that there's an FDA-approved treatment out there. The other thing I want to say is that chronically, something that was interesting is that in hypoxic ischemic injury, you get this diffusion restriction, pseudo-normalization, and then you get an increase in the diffusivity because there's just liquid replacing, or CSF replacing the tissue that's atrophic. But in these kids too, there's continued injury. So diffusion restriction continues over time as there seems to be continued injury, even in the chronic state of disease. And even though that's not going to be helpful for physicians in sort of getting timely treatment to these kids, retrospectively, it can help identify kids that were missed. So what's causing this devastating disease, and why do they have injury that looks like hypoxic ischemic injury? Well, let's jump to the underlying genetic abnormality. So in isolated sulfite oxidase deficiency, which is ISOD, and in molybdenum cofactor deficiency, the MOCOD, all of the neurodegeneration is related to the inactivation of an enzyme called sulfite oxidase, which is a molybdenum cofactor dependent enzyme. In the absence of sulfite oxidase, there's the molybdenum cofactor, by the way. In the absence of sulfite oxidase, which converts sulfite to sulfate in the final step of methionine and cysteine, and also hydrogen sulfide catabolism, you get this buildup of sulfite. Now sulfite is a really strong Lewis base. It undergoes nucleophilic attack. And so it's very toxic. And it's not something that should normally be present in your cells or floating around your body. And in fact, all of the sulfite you may take in is converted to sulfate and is excreted. Sulfite oxidase sits in the intramitochondrial membrane space, and the insertion of molybdenum cofactor is actually also important for the maturation of the protein itself. So it was thought, and it's connected to this accumulation of sulfite, and also something else I want to point out right now is called S-sulfocysteine. It's formed spontaneously when sulfite interacts with cysteine. Cysteine is the reduced form of cysteine, so it's two cysteines together. Sulfite breaks up that sulfur bridge and forms something that's called S-sulfocysteine, which is like cysteine with the sulfite on it. Okay. So the neurodegeneration that we've defined radiographically and clinically, this horrible disease that inevitably leads to death, is connected to this enzymatic deficiency in the buildup of these metabolites. The next question was, well, where is this enzyme expressed? Going back to that sort of spatial evolution of disease, right? We want to know what tissues might be dependent on this enzyme. And that could be because they use it in their normal metabolism. Perhaps it's more like a filter that breaks down some of these toxic metabolites. Where is it? My first guess was it was going to be highly expressed in the brain because that's what's affected, right? These diseases are almost purely neurodegenerative. There's almost nothing else that pops up. They don't have any other systemic ailments. In fact, the only other thing they have is really lens dislocation, where the zonules break down. And that's probably because they're high in collagen, and either sulfite attacks the collagen, or a lot of those sulfated proteoglycans within those zonules are necessary for their integrity and they break down. But we turned to mouse tissues and to human tissues to see where this protein is being expressed. And the crazy thing was it turned out to be really high in peripheral tissues. So this was like liver, kidney, and heart. And it was really lowly expressed in other CNS areas. And that was surprising because the CNS, again, is what is affected, not the heart, liver, and kidney. And these kids, they're fine. So on the left, what you're looking at is the disease relevant developmental context of P1 through 10 in or in regular normal mice. And on the right, you're looking at human tissues that were obtained through the Maryland Brain Bank. And 1, 2, and 3 refer to early gestation of 17 to 21 weeks. The middle bar is 1 to 2 days old, and the last bar is 19 to 28 days old. There's only enough 1 to 2 day old samples that you see no error bar because they had to be combined for a single sample to get all of those different areas of the brain and the periphery. But suffice to say, the surprising part here was that it's not being expressed in the brain. It almost looks like here there's not an intrinsic dependency of the brain on this enzyme. It's in the periphery. So this sets this up for like, oh, this may be cell non-autonomous neurodegeneration, this mechanism. So let's take a little closer look at, well, okay, now we know it's expressed. What's the metabolism look like? And is it really that these tissues now, heart, liver, kidney, that express this highly, are they producing sulfite? Or is it being produced elsewhere and they're really just the filters that gobble it up? Well, let me tell you a little bit about this pathway here. So sulfite oxidase sits down here at the end of the cysteine and methionine metabolism. Methionine is something that's taken up by almost all cells, and it's regulated by just your dietary intake, where cysteine is really regulated, how much gets taken into your cells. And it's super important because it sets the redox balance of your cells. It's the primary, the first step in limiting reagent in the production of glutathione. In the absence of sulfite oxidase, right, you get this buildup of sulfite. And what we knew is from just patients, actually, these are biomarkers, that there were some changes. So homocysteine levels are super low. Cysteine levels are really low. Taurine levels increase. And this molecule down here, which is called thiosulfate, is actually really elevated too. Thiosulfate, by the way, is sort of protective and not toxic. Taurine itself is not toxic and it's kind of protective too. But this sulfite is what was suspected again, and this S-sulfocysteine. Okay, so we knew about these changes. But how can we identify if these tissues, again, are really producers or not? Well, we ran into an issue here. Sulfite is really hard to measure, and that's why it had never been done before, not in these tissues. It air oxidizes really quickly, and HPLC and mass spec are actually kind of insensitive for measuring sulfite at these very low levels. So we initially ran into a technical block. We reached out and we found a dye. So this is a ratiometric fluorescent dye that was very specific to sulfite that had been reported by a group out of China. It wasn't commercially available, so I paid to synthesize it, and we got it in lab, which was great. I figured, maybe this is a starting point. The unfortunate thing was, though, as suspected, is that it was very insensitive. So you look over here, this is just a standard curve of aqueous sulfite. and you're looking at the concentrations in micromolar from 0.1 to 1. This is where we need to measure sulfite at these low levels in cells and tissues to really be able to detect it, and it just wasn't sensitive enough. But through a little bit of serendipity and a little bit of tinkering, we actually figured out a way to increase its sensitivity. And so with a few reagents, a new buffer that we came up with that stabilizes sulfite in solution, we were now able to measure sulfite down to like the nanomolar range, which was super helpful, and because we weren't able to do that before. We do hold a provisional patent on this technique, and that's important because we're trying to develop it as a diagnostic biomarker, both for prenatal detection and also for monitoring treatment efficacy. So with this new strategy, and some mentors and collaborators at the University of Cologne, this is Günther Schwarz, who's probably the leader in the biochemistry of these disorders, and a wonderful grad student that he sent to our lab to work with me for about 3 months. We decided we're going to use this to measure and show what tissues actually make sulfite. And the very first step was, because tissues were hard to come by, actually the mice weren't available to us at that time, we turned to 293 T cells that were deficient in sulfite oxidase. And indeed, if we collected these pellets using the new technique that we had, for the first time, we could show that these cells were actually producing sulfite, which is what you see here. And this bar here is when you add sulfite oxidase back, just to show the specificity of the signal. So this was a huge step forward. Indeed, the sulfite oxidase deficient cells were producing the sulfite. We took it a step further and we made double knockout cells too, just to try to understand, well, okay, great. Are we right in thinking that this oxidation of cysteine, this oxidative cysteine metabolism, is actually creating the sulfite? And in fact, it is. This is the main source of sulfite. So GOT1 and GOT2, which you also know as like ALT and AST, right? These liver enzymes, which are expressed in lots of different tissues, including astrocytes, they're directly upstream of the formation of sulfite. And in fact, what we found was that it was GOT1 that was mainly responsible. GOT2 also plays a role, but it's mainly GOT1 or GOT1 that's playing the sulfite, or the role of synthesizing sulfite in this disease. So it's neat because now it's a new target that we can go after for decreasing sulfite production in these disorders. And it also confirms our suspicion on the biochemical level of what was going on. So that was extremely powerful too. Okay, so now at this point, we have a good idea of what the disease evolution looks like. We know what the biochemical defect is. We know that that biochemical defect is leading to the production of sulfite in these tissues. And now we can actually measure it too, which is really powerful. But the next missing piece here that we haven't talked about, right, is, well, how is sulfite actually causing the degeneration in the nervous system? What's this mechanism? So for that, we turned in vitro. Everything that you're going to see here is done in cultured cortical neurons from neonatal mice, which is the developmental context, again, that's appropriate to this disease. And in the most simplistic way, all I did was take these cortical neurons, right? We just dropped disease-relevant concentrations of sulfite. And when I say disease-relevant, these are the concentrations that have been reported in the blood and the CSF of patients. So this does make sense. They're not like super high levels that are just some artificial artifact that we're creating in the lab. The very first thing I saw in these cells was that ATP levels, when you added sulfite, dropped precipitously and in a dose-dependent manner and almost immediately. And the reason that we went for ATP and started measuring that first was just because, again, the clinical picture looked like critical energy failure. And that clinical picture informed the experiments at the bench and allowed us to know what to look at. But what I'm showing you here is a little bit more too, and this was cool. It turns out, so when you add sulfite, so this happens, but it was dependent on the presence of cysteine. And this was really key because it turned out that sulfite, when you add it to media, immediately gets converted to S-sulfocysteine or SSC. And it turns out that it wasn't sulfite itself that was causing this ATP drop, because when you take cysteine out of the media, you get no drop in ATP until these like super high weird levels of sulfite. But right away, if you have cysteine, it gets converted to S-sulfocysteine and you get this effect. And so what that told me immediately was, okay, what I'm really studying in this setting here is this S-sulfocysteine. And that's really important. So I'm going to show you the model, and then I'm going to walk you through kind of how we figured it out. So what I just showed you is that, again, sulfite is being spontaneously converted to S-sulfocysteine. It's not an enzymatic reaction. It just happens spontaneously in the media, and it probably also happens spontaneously in the body as well, even though redox potentials and oxygen concentrations are different, and that can affect things. So there was something about this ATP drop that kind of smacked of like mitochondrial issues. And the reason being is that these cortical neurons are highly dependent on mitochondrial respiration for their ATP and not glycolysis. And in fact, you can take these and you can drop on, you can take away their glucose and add galactose as an inhibitor of glycolysis, and their ATP levels do change a little bit, but not drastically. If you add on oligomyosin, which is a complex 5 inhibitor, their ATP levels drop precipitously, even in the presence of tons of glucose. And so these guys are highly dependent on mitochondria for their ATP production. And it turns out you can measure mitochondrial respiration and ATP production directly in these cultures with this really cool machine called a seahorse. And what it does is, in a 96-well plate format, you have your cells, it drops these sensors into the dish and it measures oxygen consumption, which is directly related to mitochondrial oxygen consumption. And it also measures the acidification of the media as a proxy measure of lactate formation, which is telling you your glycolytic rate. And there's some other little tweaking there to determine, well, is that acidification happening because of CO2 production from the Krebs cycle versus lactate formation? You can actually suss that out. But without going into all that detail, what I want to tell you is, in these cultures, if you dropped on sulfite as sulfocysteine, or I even put glutamate in there sort of as a control, absolutely, it led to a decreased mitochondrial respiration rate, both the baseline. So what you're looking here is oxygen consumption rate. We won't go over all the details of this because it's a lot, but I think it's suffice to say that this is the basal respiration rate. This is just at rest, how much oxygen, the rate of oxygen consumption in these cells. You can see that there is a huge drop at baseline. You can also see what the maximal mitochondrial respiration rate is in these cells. So you can challenge them with drugs to drive respiration as hard as you can. And that's this bump right here in the graph. The blue is the control. And in all treated samples, you can see that maximal respiration is actually very severely impaired. So this kind of makes sense, right? These cells are dependent on mitochondria for ATP. Sulfite and it's really S-sulfocysteine in this case, right? Is dropping ATP levels and it's inhibiting mitochondrial respiration. I can also show you actually directly measuring ATP production, that it really is mitochondrial ATP production that's being decreased. So on the left, what you're seeing here in these bars, this is total ATP production rate. This is real-time ATP production. This is the glycolytic production rate in gray, and this is the mitochondrial production rate in white. And when you add sulfocysteine at 10 or 50 micromolar, again, disease-relevant concentrations found in the CSF of patients, mitochondrial ATP production really drops significantly, while the glycolytic rates, even though they're slightly affected, don't change quite as much. This is really a mitochondrial issue that's going on here. So what's causing that? Well, it turns out that S-sulfocysteine stereochemically looks a lot like glutamate. And in fact, this was reported back first by Olney et al. In I think it was like 2000 in the 90s, actually at Washington University, and they were working heavily on excitotoxicity at that time. They knew that S-sulfocysteine, because it had been known for a long time since the discovery of the disease, was elevated in these patients. And what they did was they injected it in rats, and they saw retinal injury and injury in the area postrema. And you know, that's probably because the blood-brain barrier is like really leaky in those areas, and S-sulfocysteine was making its way across the blood-brain barrier in those areas and injuring neurons. And it looked like excitotoxic injury, which was super interesting. Well, Günter Schwarz then in like 2020 showed that S-sulfocysteine, he pointed out, it looks a whole lot like glutamate stereochemically. So basically the shape and the charge distribution look a whole lot like glutamate, and that in fact, S-sulfocysteine directly activates NMDA receptors. And that's the next clue, because we you know, tons of work has gone into understanding how over or pathological activation of NMDA receptors leads to excitotoxic cell death through disruption, partially, of mitochondria. So the next step was to take blockers of NMDA receptors. And we used a ton of different ones. The one I'm going to show you is MK801. and it is a blocker of ionotropic NMDA receptors. So it actually goes in and it sits in the channel portion of the NMDA receptor and blocks the passage of sodium and potassium through there. And when we added on MK801, indeed, we were able to prevent to a large part this drop in ATP caused by S-sulfocystine. So that put the NMDA receptor right here in our model. I can also tell you that this ATP, because this drop in ATP, you could theorize or hypothesize that it's increased consumption versus decreased production. So far, what I've shown you is decreased production. To test increased consumption, the other things we did is we dropped tetrodotoxin on these cells and a variety of different anti-epileptic medications, including Keppra, phenobarb, phenytoin. None of those prevent the ATP drop. So this was really the primary cause of this ATP drop, this mitochondrial dysfunction, was related to somehow this overactivation of NMDA receptors by S-sulfocystine. What's downstream of NMDA receptors? Well, this is also very well defined, and it's calcium. So the next thing to do was just to show that it really was influx of extracellular calcium that was coming in and inhibiting mitochondrial respiration. And just by adding EGTA, which is a chelator of calcium to the media, you can see that this ATP drop in the presence of S-sulfocystine was prevented with the EGTA. So now this was super helpful because we could go back to those seahorse experiments again and either add EGTA or MK801 to see if that prevented the decrease in mitochondrial respiration. And what you can see here is the green line is treated without the inhibitors, and then the control, just untreated, is the dotted line. And the MK801 or EGTA treated ones are the blue and this sort of like camo green color. And indeed, both the basal respiration rates and the maximal respiration rates were saved by this treatment. So putting it all together, the mechanism here now, this is really satisfying because we went all the way from the enzyme, now understanding how the injury is happening in the brain, is that the molecule we suspect most is S-sulfocystine triggering excitotoxicity through NMDA receptors in the brain. Is that consistent with what we see on imaging? Yeah, definitely. And in fact, a lot of work done by, oh gosh, I forget his name, KKI was showing that NMDA receptor activation plays a huge role in neonatal hypoxic ischemic injury, and that probably explains why this is such a beautiful phenocopy. The other thing I want to say to you, though, is this is in vitro, right? This isn't perfect. And indeed, sulfite itself, Anna and I published another paper showing that sulfite actually does disrupt the mitochondrial network architecture that leads to hyperfused mitochondria that are still respiring, but actually not quite as well as you would think. So there's actually probably a dual mechanism here too between both sulfite and S-sulfocystine. Okay, so now we know this mechanism, we have a more understanding of the disease pathology. How are we going to develop a gene therapy for these guys? Well, we need a mouse model. So we turn to MOCS1 knockout mice. And here's something else I want to point out. These mice weren't available. I was only able to get these through a sponsored research agreement with Origin Biosciences. So this interaction with industry became very necessary. And actually, they also sponsored a lot of this work because I couldn't afford to do it. So this was really helpful working as a team. So these mice, the pathophysiology actually nicely recapitulates what you see in humans. And their growth curve it falls off around day 5. Survival, they start dying around day 5 and they don't survive past day 10. And when you look at them clinically and do mouse neurology on them, there's a lot of different battery of studies you can use. But two of the ones that I think were really helpful were hind limb suspension and the writing reflex, which really showed a deviation from controls around the same time that the growth curves were dropping off and that these guys were starting to die. So there's this pre-symptomatic period, and then there's the development of symptoms, and we can measure this clinically very easily, which is awesome. I'd also like to point out that these mice, it was known before because they had been partially characterized by Günter Schwartz's lab, that they also biochemically reflect the human condition with lack of molybdenum cofactor production, elevated S-sulfocystine in the urine. And then the other thing that we found, which was cool, which wasn't known at the time these mice were first created, is it turns out, again, I mentioned this earlier, sulfite oxidase needs molybdenum cofactor just to mature and be present as a protein. So if you look at the liver, kidney, and the brain of these mice, brain doesn't really have sulfite oxidase in it that much, but look at the liver and kidney. These are two wild types they have tons of sulfite oxidase, but sulfite oxidase protein is missing in the molybdenum cofactor knockout mice because the protein can't mature without the cofactor. This is cool because when thinking about gene therapy, this gives a functional readout for the efficacy of molybdenum cofactor production in these mice. Not only are we just showing that we delivered a transgene, but if we can restore sulfite oxidase protein levels, that's a downstream functional readout that our treatment is working. So now we have also a biomarker of treatment efficacy that we can use. But it turns out molybdenum or MOCS1, the gene, is actually extremely complicated. It is one of the few bicistronic genes that we have as humans. You know, you learn in like college biology that polycistronic genes or mRNA only really exist in prokaryotes, but it turns out there's some bicistronic genes that do exist in eukaryotes, and this is one of them. So it's one gene, one gene, and through differential splicing produces actually totally different proteins. And it was through an incomplete fusion within evolution that this happened. And I just want to tell you briefly that we came up with two different constructs, one that had 2 introns in it, one that had 1 intron in it, and then we started manipulating, or we are manipulating intron size, and we're able to actually recapitulate or reproduce all of the necessary proteins. So there is a fusion protein that we're able to pop up. There is an active MOCS1B that is an enzyme that's really important for creation of molybdenum cofactor, and also this MOCS1A. And I just want to point out that by putting an intron upstream, we can actually change the ratios too. We can start to manipulate how much A versus B we get. And we're making these smaller and changing them up to make them clinically applicable and packageable into AAV vectors. And these things work. So check this out. This is in the 293 cells. These are MOCS1 knockout 293 cells. And what I want to show you is that these vectors can restore sulfite oxidase in these MOCS1 knockout 293 T cells. Now, they don't make, 293 cells don't make a ton of sulfite oxidase, but basically the gist of it is, so this is with empty vectors. So this is the level of sulfite oxidase in the knockout cells. If we put in an inactive MOCS1 control, this is one of our vectors, but we've inactivated the enzymes, no change. But if you put in our 2 and our 1 intron constructs, these gene therapy constructs, it actually restores sulfite oxidase levels pretty nicely, actually almost up to the wild type levels. So in vitro, we have working constructs that we're now going to be putting into the animals. But you can probably see some of the challenges here, right? Let's go over the model of what the disease is and thinking about moving this to a first in human application. So what do we think is going on? All right, so with all of the data I've given you, here's what the disease model is. In utero, right, and in life, liver, kidney, heart, these peripheral organs are producing this sulfite. The sulfite gets into the blood and is probably turned into S-sulfocystine when it interacts with sulfite. These kids are born pretty normal. And so it turns out that these abnormal metabolites are being filtered from the fetal circulation by the placenta and the maternal circulation. So in utero, they're fine. They're born, and there's a very short pre-symptomatic stage. That's your therapeutic window to intervene and do something to save these kids before there's this severe onset of neurological dysfunction and they head downstream. Now, one could say, well, we could do gene therapy right at the onset of this, but it takes time, right? Like the vector has to get there, it has to express, you have to like identify these kids and diagnose them before you can do this. And that's probably going to preclude the ability to actually do gene therapy in these kids if you didn't know ahead of time that they already had it. So if you have a sibling that has it and then you test the kid, that's going to work. However, now that we know the mechanism about what's going on, we've started to use NMDA antagonists like memantine to prevent the brain injury and widen that therapeutic window. Fostinopterin, which is that's the drug that was FDA approved for MOCS1 type A. We're also trying to use that to broaden the therapeutic window. That'll only be applicable to that subtype of patients, but memantine and other NMDA antagonists will be broadly applicable to all of these kids. And that will allow us, hopefully, to broaden this window for intervention. Now, it's really hard to develop these things, but through Origin Biosciences, we were able to find a partner that is interested in licensing these gene therapies and actually helping us build these therapeutic strategies with memantine. So that's awesome because we have the open door to walk through and we just have to build the therapies. So hopefully that gets to the patients soon. They've also done a wonderful job just building up the patient-physician awareness. I've been working with them and educating people and getting that out there. So I think that the field is ready for these sorts of treatments, and that's exciting. All right, quick change at the end of the presentation here. This will be a lot shorter. Mucolipidosis type 4. Now, this is a seriously cool disease. This is a lysosomal disorder. Rather than a lysosomal storage disorder, it's a mutation in the gene that is a cation channel in the lysosomes. I got into this project when Florian introduced me to the ML4 Foundation. So we're going to switch gears here. ML4 Foundation is the only international patient advocacy group for these kids. And they've done this amazing job of organizing like over 100 patients worldwide. They're motivated to fundraise. They're motivated to get these kids treated, and the families all want to participate in research. In order to do translational research, it has to be a team effort. And you can see all of these amazing people down here that have been working with us to really get this project off the ground. These kids are all over the world. There was a higher prevalence in the Ashkenazi Jewish population, but because of preconception genetic screening, the incidence is actually going down within that population. And now most of my kids are from all over the world. I've got kids in the UK, South America, Australia, France, the Middle East, everywhere they're popping up. It's just higher patient awareness. But through this group, again, these kids are, they're very fun. They have a great personality. They love music. And this population was just basically ready to go in terms of getting treatment to them. And let me tell you a little bit about the disorder too, just to set the stage. So this is mostly a CNS disorder. And what we had known about it was really just from case series and case studies, and that this was like a developmental disorder with a plateau in development where kids peak around like, maybe like the 18 to 20 month old level of gross fine motor and intellectual function, but not too much else was known about that. They do have corneal clouding. They have very rapid retinal degeneration. They have achlorhydria, which results in iron deficiency anemia. They may have some kind of renal thing going on, which I think is real. Some have acute on chronic renal failure in the second decade or third decade of life. And then there was a question about proximal muscle weakness that I don't think plays a big role in the disease in humans. And just because we're all neurologists here, probably interested in seeing the brain, it also turns out this is a hypomyelinating leukodystrophy with brain iron accumulation in the basal ganglia. And that was kind of cool because after seeing these kids, we traveled to Israel, saw some kids there, traveled around the United States, saw some kids here. It was amazing. We were actually the first group to describe extrapyramidal signs in these kids. So it turns out they have a Parkinsonian-like disorder that hadn't been appreciated before, with bradykinesia, rigidity, and some tremor, as well as other signs of spasticity, upper motor neuron weakness, and dysfunction. There's a long legacy of this disorder here. Back in 2000, it was Dr. Sue Slagenhop, who's just amazing scientist, was able to clone and identify the gene that was responsible for this disorder. And it was named MCOLN1, which encodes this cation channel, the protein, which is called TRPML1. And then another amazing scientist, Dr. Yulia Grishchuk, characterized the animal model and came up with a working gene therapy, preclinical gene therapy that looked just so promising. And what was really cool about this is that these mice, it didn't just prevent progression of disease, it actually improved function. It looked like it's restored neurodevelopment, which was really fascinating. And the reason behind that probably is because this isn't a neurodegenerative condition early on in the first decade, right? This is the opposite of sulfite oxidase. We have a huge therapeutic window where neuronal populations are still functional and viable. And if we can deliver this gene to them, we can kick them back on and restore neurodevelopment. But what was missing in this case, and here's the story here, is that natural history was missing. So we had patient advocacy, we had the science, but we didn't have that natural history to help bridge the gap to that first in human application and our IND. So working with Yulia, we were able to see, and the ML4 Foundation, we went all around the world seeing these kids. And I won't bore you with the data, just suffice to say that we were able to quantitatively define the increase in spasticity and rigidity, the decline in finding gross motor function, and identify the threshold of age over which kids start to decline. And really that led to this disease model where, again, there's this plateau in development for the ML4 kids in the set of this worsening visual impairment, and then an eventual decline in function as the neurodegenerative component kicks on in this third stage of disease, which is in like the adolescence. And our hope for these kids is that the neurodevelopment or the treatment will kick back on neurodevelopment like it does in the mice. And that'll be exciting. That's a paradigm shift, right? This is gene therapy to restore neurodevelopment, not just to stop neurodegeneration. So that's moving forward. The other thing that we needed was a biomarker. So in collaboration with Levi Wood at Emory University, we were able to collect samples. I collected from both parents and kids. And it turns out, informed by Yulia's work, that there was a CNS inflammatory reaction in the mice. We went and we defined basically a set of cytokines and interleukins that were elevated in patients and also corresponded with severity of disease. And we did this in the mice and then in the humans, but in the humans, we didn't have CSF. So the mice allowed us to, there's fancy looking correlations, look to see in the mice if we could predict what peripheral cytokines predicted CNS cytokines, which of those predicted disease progression, and which of those correlated with the humans. And this sets us up, it's like hypothesis driving now too, right? Now we're going back to the patients and seeing if we can figure out and correlate these things with disease progression. And in the mice, see if we can use gene therapy to affect progression and then see if the biomarkers change. And this is really important because a biomarker for a disorder like this, where you don't have other great readouts, only clinical readouts, and which also may take time to show improvement in function in clinical readouts, these biomarkers might be fast proxy measures for showing treatment efficacy. And this is a really important part of moving forward. So what I want to say is just translational research is a team effort, and I have so many people to thank for the support and everything. I really want to say thank you so much to Florian Eichler for being my mentor, and Kevin Staley, and also my career mentors, Merit and Sue Slagenhop. Thank you so much for all of your support. My funding from the NIH and Origin Biosciences, the Schwarz Lab at the University of Cologne, who's just been also mentors to me at this time, all of my mentees. And then on the ML4 side, there's a huge team. A huge thanks to Gabriella, Pat, and Tim, who have been driving from Partners Innovation towards getting this developed by industry, because we really need that partnership to produce the product and get it into patients. Yulia and Sue, and then all of the clinical members from around the world who've been helping with this. And I just made this opportunity to thank all of my clinical mentors that have made me the physician who I am today, that have allowed me to do this. Vern Cavanis, Dave Holtzman, Dr. Dooling, Robin Jones, Kevin, Florian, everyone, Dr. Teal, thank you so much for all of the teaching over the years. And thank you to the department for all your support. Great.
Thank you, Albert. I wanted to just say a few words. First of all, your work is so amazing and important. I really feel inspired by your work that you're going to find treatments for these rare and really serious disorders. So kudos to you and your amazing team. I know you spent a lot of time on building natural history and really thinking creatively about how do you do studies in the rare disease. I was just wondering if you had any pointers for maybe other people in the audience that have that interest of how do you actually organize and get that done? How important is it for what you're doing?
Awesome. Yes, super important, and different challenges with different types of disease progression. Some of the most devastating disorders that are in clinical trials now, like Tay-Sachs-Sandhoff's and those Canavan's disease, the retrospective studies are, so that's definitely important, because that can give you a huge population base very quickly. Those can be a little bit more facile or easy if it's really devastating, because even just survival might be enough to drive the natural history, give you biomarkers. But in other disorders like ML4, which are slowly progressive, huge challenge. And so the approach was, for us, it was multimodal. So it was a cross-sectional natural history study to get an idea of what's going on and really inform our understanding of the disease, moving to a retrospective study that's limited by just the intrinsic limitations of retrospective medical record review, but it gives you lifetimes of data, right, that you can review and sort of understand. And we tried to push a little bit more by, we designed our own scales for ML4 based off of our cross-sectional data, and we made those robust so that they could be applied retrospectively as well as prospectively, so that we can quantify sort of progression of disease in the medical records. And then we pushed to a lead-in study. So we also are now trying to push for a prospective natural history study, not in the sense that we want to prospectively follow kids over years and years and years, because we don't have time. The treatment's ready and these kids are getting worse, but rather define the baseline for these kids, compare it in the context of the retrospective study, and then show what happens after treatment. So it's kind of a neat paradigm, right? Because we almost jump to the treatment because they're monogenic. We know what to do right away, even before maybe you know what the natural history is, but you have the impetus is there to treat. You also have the moral imperative to do something because you can, and these kids have nothing else. So it's a new technique. Right? It's sort of building the natural history around the treatment itself. And in fact, I think this is kind of the future too, by the way, is this new reverse translational studies that are going to happen where you treat the kids and you've created a new biological system by doing so. And then you have to study what you created to understand the disease and the treatment itself.
Thank you so much, Albert. We're going to now open up a question and answer portion of the presentation. We have one question from an anonymous attendee. How do you ensure your study populations are heterogeneous for ultra-rare disorder studies?
Oh, sorry, what was it again, John?
How do you ensure your study populations are heterogeneous for ultra-rare disorder studies?
Yeah, okay, so there is a lot of heterogeneity among these populations, which can be really challenging. In the case of sulfite oxidase deficiency in the typical kids, it's such a devastating disorder that really the phenotypes almost converge because they're so severe, they almost all look the same. ML4 is the opposite, right? There is heterogeneous populations across these kids, and that can be really difficult because the cohort analyses that you would have to do with a lot of variation required high numbers, and that's not possible if there's not a lot of patients. So what we've turned to the technique is actually these parallel N-of-1 studies, and that's how we've kind of been thinking about this. Oncologists, I think the field of oncology are light years ahead of us, and that they're already there. They're to this stage of precision medicine, where they are practically doing these massive in parallel N-of-1 studies that are successful because they're embedded within these new types of clinical trials, like platform trials, umbrella trials, master observational trials, that are also defining these populations by biomarkers and characteristics, and understanding how that variation affects the outcome of treatment. And if you do this in an intelligent way like that, like they're doing that, you actually can run tons of N-of-1 trials and infer effectiveness across all of these patients by all of these different factors that you're following, and also just designing how are you going to tell if they affected these individual patients. So can you learn something from N-of-1's trials? Yeah, absolutely, but you got to be smart about it. There's a whole consortium that's built around this. It's called the N-of-1 consortium. And in fact, places like Stanford are pouring millions of dollars into this to build structures where you can have, let's call it, we have genomic medicine or medical genetics, but the next phase clinically is going to be interventional genetics, where we're actually taking these patients, seeing them in clinic, and then doing something. Maybe it's delivering a transgene, maybe it's CRISPR editing and doing something immediately to help these patients, and then learning from them, building them into these higher organizations of platform studies and master observational trials to make sure that we're actually learning what we're doing and how to move forward from there. So I think all of this is moving, field of medicine in general is moving towards precision medicine, and this is just another way of how that's happening.
Excellent, thank you. From Patty, she says, could you comment on how you work with the FDA on the N-of-1 studies?
Yeah, they're becoming more open to that. So that's a great question. From an academic perspective, they are, I think a bit more open to investigator-driven trials like this. And if you have a product that they understand, so they're understanding more of the safety around ASOs, they're understanding safety around the AAV vectors and that sort of thing. And it's really that you have to show some of the safety around whatever specific transgene you're showing, or maybe the sequence of the ASO. The challenge there is that you do need tox data though, right? Like the standards for non-human primate and for rodent toxicology are not going to be very different if you're doing an N-of-1 versus an N-of-20. They're still going to want to see it. They're still going to want to know it. And that takes money. Non-human primate studies are expensive. We're talking like $46,000 a monkey, and you need maybe 6 to 12 monkeys or something like that. So money is a barrier, but if you have the resources to do it, it's absolutely doable.
Can you, this is from an anonymous attendee, could you please explain the bicistronic expression of 2 isoforms of one gene using one construct?
Oh, yeah, definitely. So 2 cool examples of that. So with the MOCS1, what's happening there is, I don't know, can I go, are my slides still showing?
They are not.
Okay, that's okay. So basically what happens with that one of the constructs, the MOCS1, is that there are two different splice forms. One of them produces a fusion protein where there is MOCS1A and MOCS1B in tandem, but there is a stop site in between the two, so only MOCS1A gets expressed. And then in the second case, there is a splice variant where that stop site gets cut out. And then you get this fusion between MOCS1A and 1B, but A is actually inactive because as well as cutting out that stop site, you've also cut out a double glycine catalytic site that's necessary for it. So you have this weird fusion protein where it's inactive A and active B. And what's been shown by Günter Schwarz is actually then that gets further processed because it has a mitochondrial import signal downstream of it and a cleavage site where that inactive A gets cleaved off and you just have active B. And what probably happened here evolutionarily is that there was an incomplete fusion event that for some reason persisted for millions and millions of years, probably because there's some utility to it that we don't understand, but that's how that one is being driven. And then we're just messing with the intron sizes and positioning of the introns to affect how much of one versus the other splice product is being produced, and then trying to understand how that affects the efficacy of restoring sulfite oxidase function.
I think we've got time for one more question, so I'll finish with this. What would you dream of having at MGH to enable these types of studies from a regulatory and statistician support?
Oh gosh, oh, so many things. So yeah, I mean, this is sort of the thing. Like, you know, academia is such a wonderful place to do basic science, probably the best place. It's an amazing place to run clinical trials. But translational science like this can be really difficult because I think the incentive system, and you know, this is just across the board in academia, isn't really set up to reward or promote team-based approaches, right? So like, how do you get forward in academia? First and last author. But if you're a team of 10, can't all be first and last authors. So it's a little bit harder to get buy-in to that. Also, a lot of these studies, like natural history studies, these are not sexy. This is not what the NIH wants to fund, right? This is, you gotta find funding in a lot of different places. Our natural history studies and all the work we did, zero NIH funding. This was all from like small foundations, from donations. You know, Rebecca Overman, who's the head of the ML4 Foundation, doing amazing work, like getting us this money. But what we need really is an infrastructure that sort of mimics industry in some ways, where you have people that are all going towards a common goal and that have different specialties and are all working forward towards, you know, making this treatment. So biostatisticians, a regulatory consultant, the scientists and the clinicians coming together to do the research, a mechanism of funding, or, you know, coming from industry does fund this stuff. Like you can find SRAs, you know, in different companies that are interested in funding this, foundations and small foundations that are willing to fund this, and donation. And so if you have all of that working together and forming sort of this, for lack of a better term, like mini industry sort of approach, I think that's what would be really successful. And that's what would really help us push forward.
Awesome. Thank you so much, Albert.
That
was fantastic.
Thank you guys. Awesome. And thanks again for this opportunity. It was so great.
Have a great day.
Thanks. Bye, guys.